Hello everyone, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we are going to be learning about viruses. They can infect everything from bacteria to human beings and everything in between. They've been known to infect every type of living organism. So let's go take a closer look. So first things first, viruses are very small. At least most of them are. Yes, there are some giant viruses out there that are almost the size of bacteria, and you can actually see them using a standard light microscope. However, the vast majority of them you can only visualize using an electron microscope, and the most common ones that you see are usually more along the size of a Zika virus or a parvovirus. They're going to be absolutely tiny. Another thing to consider is that viruses are obligate intracellular parasites. In other words, they need a living cell in order to reproduce. Pretty much the only thing they're going to bring is some genetic material, maybe a couple enzymes, but they rely on cells for just about everything else, for energy, for raw materials, for building new viral particles. They really are dependent upon those cells. Now, we can classify viruses in a number of different ways, and one of the ways that we classify viruses is based on their shape. And so we've got the spherical or polyhedral, sometimes called icosahedral or isometric. They look more circular, almost soccer ball-like. And then you've got the complex viruses here, like the bacteriophage. These are viruses that only infect bacteria. They're kind of like space aliens. And you've got the helical viruses. And not shown here, you have some irregularly shaped viruses. Ebola virus would be an example. And those are called pleomorphs. And so here you can see that Ebola virus, that pleomorph, where it looks like kind of like almost like a piece of spaghetti noodle. The other way that we classify viruses is based on their genetic material. They can be single-stranded or double-stranded, DNA or RNA. And what's interesting about viruses is, yes, some of them are single-stranded DNA, some of them are double-stranded RNA. And so what we've learned in the past about double-stranded DNA moving to single-stranded RNA, that's not always the case when it comes to viruses. What's interesting about viruses is they have DNA or RNA in the capsid, but not both. Where living cells, they always have both. They have a DNA genome and they have RNA. So there are two main types of viral infections. One is called productive and the other is latent. In a productive infection, the virus is gaining entry into the cell. A single virus can often give rise to hundreds, maybe even thousands of viral particles from one infected cell. Latent infections are a little bit different. The virus enters the cell and then goes into a sort of dormancy. And we're going to talk about how that happens here in a second. But in a latent infection, the cell is not actively producing viral particles. So I wanted to start off a little bit on the simple side here when it comes to how viruses actually replicate. They have two different cycles that they can go through. One is the lytic and one is called lysogenic. And what we're focused here is on viruses that infect bacteria. In a few minutes, we'll contrast this to how viruses infect animals. But let's go ahead and start in the upper left-hand corner. We've got a couple of bacteriophages there, 3 to be exact. And you can see here that the bacteriophage, the first thing it must do is attach to a host cell. And in fact, when it comes to pathogenesis, a pathogen, the first thing it must do is always attach to its host. If it can't attach to the host, it's probably not going to cause an infection. So attachment is thing number one. Next step is you're going to see that the virus injects its genetic material or releases the genome into the cell. Step 3, and here we see on the left-hand side, bottom left, we've got replication. And what's happening here is there's actually a few things happening at this step. The virus is going through replication of its genome, but it also has to transcribe its genome and then translate that RNA into proteins so that we have all the pieces necessary for viral assembly, which is the next step. And so if we look at it so far, the virus first has to attach. Second, we have genome entry. Then we have replication, transcription, and translation, not necessarily in that order. That is virus dependent, but replication, transcription, translation, all happening. Once that happens and we have the raw materials, the virus can then assemble. And what that means is the genetic material is surrounded by that protein capsid. And then finally, the virus will release and lyse the cell. It's important to note that once a virus gains entry into the cell, it usually takes that cell over. It now commands that cell, and the cell is basically a slave to the virus. Now, there is another potential option here, and that's the lysogenic cycle, and that's what we see on the right-hand side. Well, now what's happening is, yes, you have the bacterial virus that still has to attach. We still have the virus genome entering the cell. But then if you look towards the bottom, we get what's known as integration. The viral genome integrates into the host chromosome, and you can see the two colors are mixed now. Now, this integration, when that happens, if it is, like we're talking about here, a bacteria virus, we call that a prophage. And so when you see that designation pro in front of either phage or virus, that means that the viral genome is incorporated into the host genome. You're going to notice now here at the bottom right, we have a couple of options here. Option number one is that the genome stays integrated. And at this point, it would be nearly impossible for the immune system to recognize that this is happening. The virus can technically replicate, and the way it does that is every time the host cell replicates, the virus genome is replicating with the host genome. And so as you can see there, that vertical gene transfer, as the bacterial cell is replicating, it's also replicating the viral genome. And it can stay that way for an undetermined period of time. And then what can happen is the virus, sensing that the host cell is experiencing some type of stress, it can go back into a productive infection, at which time the viral genome will release itself from the host genome and go back into replication, transcription, translation. That's on the right-hand side there, assembly, and then release. And so this lysogenic cycle is really interesting because essentially we have the lytic cycle, we have attachment, genome entry, but then we have this intermediate step, and that is viral genome incorporation. And it can stay that way. Now, in human beings, when this happens, usually what happens is there's some type of stress in a cell. And so we could use the human herpes virus as an example. And so what can happen is the virus causes an outbreak, it's replicating, the immune system targets the virus, causes damage, local damage to the epithelial tissue around where the virus is releasing, and so that can cause a cold sore on the mouth, and then the virus retreats. The immune system doesn't completely clear it. The virus retreats into other cells, goes into this state where it is undetectable by the immune system. That wound heals up, but the virus is not gone, and then a stressor can allow the virus to begin replication again, and there can be a reoccurrence. And that's why for cold sores, they can go away and come back periodically. All right, let's review in a bacterial viral infection, the lytic cycle. The first thing is attachment, then genome entry. Then you get replication of the genetic material along with transcription and translation so that the capsid proteins can be made. Then there's assembly and then release from the cell. Now with animal viruses, viruses that infect, let's say, human beings, it's a little bit more complicated. We have a few more steps because we're dealing with a eukaryotic cell that's going to be larger, compartmentalized. There are specialized areas within a eukaryotic cell where certain functions can happen. And so when a virus gains entry into a human cell, things are a little bit different. So attachment is still the first thing that has to happen. However, in viruses that infect humans, it's not just the genome that's entering the cell. Usually it's the entire capsid with the genome still inside. Because of that, there are two additional steps that happen. One, the virus has to actually go to a site of replication. As an example, let's say you have a virus that is double-stranded DNA, and the virus needs to replicate that double-stranded DNA. Well, where's it going to go to do that? Where are the enzymes located in a eukaryotic cell that allows the virus to replicate double-stranded DNA? If you said the nucleus, you would be correct. And so that particular virus is going to have to target to the nucleus. Not all viruses do that, but in this particular example, that's where it would have to go. The virus also has to uncoat. It has to get rid of that capsid and release the genome so that in the next step, replication, transcription, and translation, all of that can happen. And of course, once that does, you're still going to get viral assembly, and so you're going to have that protein coat for the capsid surrounding the genetic material. But then here's where things get a little bit different again, because as the virus leaves, right, so causes cell lysis or buds from the cell, In this particular case, now the virus is not in the environment like it was for a bacterial infection. Now it's in the host. And so it actually has to avoid the host immune response and spread within the host. There's an added complication there, and now the virus has to eventually exit the host, get out of the host, and find a new host, because what usually happens, hopefully for us human beings, is our immune system fights the virus and clears it. So the virus is always racing against the human immune system. The virus has to get in, do all of its replication, exit the cell, get out of that host before the immune response happens, and get into a new host. And so when it's shedding from the host itself, that's often when we're sneezing or coughing, when there is bodily secretions happening that is shedding that virus, making it more likely that the virus will find a new host and do the exact same thing again. Something else to consider when it comes to viruses exiting the host cell, they don't always lyse the cell. And so that will often happen in a lytic infection. But the virus, especially in animal viruses, sometimes they release from the host and they actually become enveloped. They take a piece of the host membrane with them, and that's an envelope virus. They're enveloped with the host membrane. And so just want to make sure we understand that not all viruses completely lyse the cell. Some of them are a little bit more civilized. They exit the cell, take some membrane with them, and usually the cell is going to die, but it's not necessarily going to die via cell lysis as quickly as a virus that's just going through lytic cycle. One enters, hundreds or thousands leave. Now, what's really interesting about viruses is viruses have what's known as a host range. And that host range is primarily determined by two major factors. Factor number one is, can the virus attach to a host cell? Can the virus attach to a cell? The virus has very specific attachment proteins, and it uses those proteins to attach to a host. If the host lacks the attachment proteins that the virus requires, well, guess what? The virus can't attach, and we said that was the very first step in a viral infection. So no attachment, no infection. But even if the virus can attach, and let's say for argument's sake it actually even gains entry into the cell, the cell, the host cell, still has to be permissible to viral infection. Well, what does that mean? It means that the host cell must have the enzymes required by the virus in order to replicate. And without that, even if the virus were to attach and gain entry, if the host cell is not permissible, in other words, it doesn't have the machinery that the virus needs in order to replicate, there isn't going to be any replication or viral infection. Now, if there is a viral infection, that can look a few different ways. The first way is there could be an acute infection. A really good example of that would be the common cold. The virus gains entry into the host, it starts to replicate and do all the things we just talked about. Eventually, the host starts exhibiting signs and symptoms. They usually come on within a few days, maybe a week, maybe slightly longer, depends on the virus, could be as long as a couple weeks. But during that time, now the host, let's say it's a human being, is starting to feel those symptoms. Also, the host will start to show the signs. And so maybe if it was influenza, you might have a fever, there might be a cough, might not feel all that well. Well, that will stay that way for a number of days. And then there is a recovery period. Generally speaking, for acute infections, they might last a week, maybe a couple weeks. There could be some symptoms or signs that stay on and last a little bit longer, but for the most part, acute infections tend to be relatively short-lived. The next type of infection is a latent infection. We've kind of discussed this already, because in a latent infection, usually that's analogous to a lysogenic infection. So there's an acute phase where someone is showing the signs and feeling the symptoms, and then it seems to just go away. But like we've already talked about with the oral herpes example, that cold sore can appear. The immune system seems to fight it back, but it goes into hiding. At that point, we're in a lysogenic prophase, or in this case, provirus phase of infection. The immune system doesn't know it's there. Everything seems to be okay, and then it can come back. And so that's what happens in a latent infection. Another example would be chickenpox. Chickenpox early on in someone's life can then turn into shingles later on in life. It's why the vaccine is so popular, because it prevents that shingles usually from coming back later on in life, because the person never got the chickenpox in the first place. Then there is the chronic infection. And with a chronic infection, this is a virus where the viral particles elevate, but they never actually are reduced like you would see with a latent infection. We have an acute infection, the viral particles seem to go away and then they come back. With a chronic viral infection, you have that escalation in viral particles, that increase in viral particles, but it stays fairly consistent or could even build a little bit over time. A really good example of this would be certain types of hepatitis, where what winds up happening is the virus remains elevated for a long period of time, and then over the course of months or maybe even years or longer, that's when the individual really starts to show the signs and feel the symptoms. But the viral load never really decreases. It kind of goes elevated and it stays that way. And then finally, we have the slow infection, and this was really a term that was mostly invented for HIV infections. And initially, HIV infection looks like an acute infection, and it appears as though the immune system does what it does with a common cold or an influenza virus. It beats it back, but as it turns out, the virus never fully goes away. It winds up infecting more immune system cells, but then over time, the viral load increases. And so it's different from a chronic infection because the chronic infection, you have that acute phase and the viral load stays consistent. With the slow infection, it almost looks like a latent infection where you've got the acute phase, it goes all the way down, but then over time you start to see an increase in the number of viral particles. At some point, as the viral load gets too high and the immune system is being destroyed, that is usually when somebody starts to show the signs and feel the symptoms. Now, there are certain types of viruses that can actually cause cancer. An HPV, human papillomavirus, has been one that has been associated with cancer. Why? Because HPV can be around in the body a long time, and it is a double-stranded DNA virus. And like we talked about already, double-stranded DNA viruses, they need to go into the nucleus, or at least turn on the machinery that's inside the nucleus so that they can replicate their genetic material. Well, think about it. If you have a cell that is not normally replicating and it receives a signal, which is essentially what the virus does, is it tells the cell, the host cell, hey, I need you to turn on DNA polymerase, and I need the primase, and I need all these things, right? The helicase, the ligase, all those things we talked about when we were discussing replication. Sometimes what that triggers a cell to do is to start to replicate as well, and that is when cells are replicating and they're not supposed to be, that's cancer. Another reason why viruses may cause cancer is they may insert themselves when they are a provirus in certain areas within the genome that is responsible for controlling cell growth. And if those areas get disrupted, you can wind up with a cell that now starts to grow out of control. Now, we briefly already mentioned HIV, and HIV is a very specific example of a virus that I want to spend a little bit more time on. It is a slow infection, and so HIV, there's an initial acute phase, and then it just seems like the immune system destroys all the virus and all the cells infected by the virus, but that's not exactly what happens. And over time, the virus hangs out and destroys more and more immune system cells until somebody starts to, once again, show the signs, feel the symptoms. And so HIV is a unique virus because it's what's known as a retrovirus. Retroviruses are named because they actually have an enzyme called reverse transcriptase. And so retroviruses with reverse transcriptase, what they do is they start with an RNA genome. And that RNA genome is then reverse transcribed. We go from RNA to DNA. It's backwards from what we've learned so far. Human beings, all living things, they go DNA to RNA to protein. We've already discussed that. Retroviruses start with an RNA genome. They use reverse transcriptase to make a complementary strand of DNA. And then that same reverse transcriptase is responsible for taking that single strand of DNA and making it double-stranded. Now, there's a problem with reverse transcriptase, and that is it has basically no proofreading ability like DNA polymerase does. And so what you wind up with are lots and lots of viral particles that are mutated. In fact, if you were to look at a single individual and look at all the single individual that is HIV positive and look at all the HIV particles, viral particles in their body, you could theoretically have a mutation at almost every single point in the genome. So lots and lots of genetic diversity within a population of HIV viral particles in an infected person. That presents some very serious issues when it comes to trying to create a vaccine because vaccines typically train the immune system on how to recognize certain proteins that are associated with viruses or bacteria. Well, if the HIV particles are constantly looking different from a genotypic standpoint, the genes are different, that's going to lead to a phenotypic difference. The physical characteristics, the proteins on the capsid, they're going to look different. And just when the immune system starts to get used to seeing one type of protein, the virus looks different and it can't possibly keep up with all the changes. So HIV, it's this retrovirus. It has reverse transcriptase. Once it makes that double-stranded DNA is incorporated into the host genome. Sound familiar? That's a lysogenic cycle. There's a certain enzyme responsible for that, and that's called integrase. And integrase is really, really interesting. It turns out the more people study HIV, the more they realize HIV is actually really good at inserting itself in an area of the genome that's not going to disrupt the cell too much. And it's really consistent about how it does it. And so HIV can lay dormant in immune system cells and other cells besides those of the immune system. And then what happens is there is some type of trigger, there is some type of signal that tells HIV to begin going through the lytic cycle again, which is replication, transcription, translation. It will assemble and then it will leave the cell and infect other cells. The other thing that's interesting about HIV is when it goes through the replication and especially the transcription translation, it usually makes one big long messenger RNA transcript. And that messenger RNA transcript makes a really large polyprotein that has to be chopped up into usable pieces so that the HIV can actually form a capsid and also have the enzymes that it needs, like the integrase enzyme and the reverse transcriptase before it goes. Now, what's interesting is we have learned that because HIV relies upon reverse transcriptase, relies upon integrase, relies upon certain proteases that are responsible for chopping up this polyprotein, we can make medications that target these enzymes that slows or even pretty much stops HIV replication in a person who is HIV positive. One of the things that viruses can do, and it is a real annoying thing for human beings and quite frankly downright dangerous, is viruses can change their proteins. We saw that, we talked about it already with HIV, because reverse transcriptase has basically no proofreading ability. Well, that is basically the same thing for viruses that have other types of RNA genomes where they're not retroviruses, but they use an RNA-dependent RNA polymerase to copy RNA into more RNA, something that we human beings cannot do, but certain viruses can. And so it can happen over time, and this is certainly true of influenza, is influenza, as it is making new proteins, new capsids, a lot of times you'll start to see some changes in those proteins and just a slow, what we call genetic drift or antigenic drift. And so the virus begins to look a little bit different than the original strain. And this is constantly happening. And it's the reason why a new influenza vaccine is released every year, because as we get the influenza virus up in the northern hemisphere in our winter time, the virus is going through antigenic drift slowly, but then it goes to the southern hemisphere when the northern hemisphere is experiencing warmer temperatures and we're getting more drift. And then what scientists are trying to do is figure out, okay, what are the most predominant protein types on the capsid of the influenza virus, and they make their vaccine based on that. And so antigenic drift is a slow, methodical, almost methodical-like change in the proteins of a viral capsid. Now, antigenic shift is a potentially far more problematic occurrence. And so what we have here is, let's say we've got influenza virus A and influenza virus B, not to be confused with the actual strains, but let's just say we have two different variants of an influenza virus. Let's say virus A infects human beings, virus B infects birds or pigs. And let's say both of those viruses happen to gain entry into a single cell. Doesn't matter if it's a bird or a pig, but they both gain entry. What can happen in antigenic shift is they get a mixing of genome in a single capsid. And that single capsid is the new strain that we see on the far right-hand side that now shares proteins between virus A and virus B. That new strain may potentially be able to infect a new organism. It has increased or changed its host range. And so what can happen, especially with influenza, especially with avian or bird flu strains, is they can go through antigenic shift or drift, but shift is the one that we are talking about here. And now they gain new potential to infect new hosts. And often this has the potential to cause pandemics. And so with this, what we wind up with is a virus that now has a new host, can cause new infections, but unfortunately the hosts that are experiencing this new infection don't really have any herd immunity, and their immune system really hasn't seen this virus before, and so it could potentially spread very quickly before the human immune system kicks in. One of the things I find very interesting is bacteria, just like human beings, have mechanisms that protect themselves against viral infection. And one of those mechanisms is something called restriction modification. Now, what I'm showing you here is restriction modification used in biotechnology, but originally its natural purpose is to protect bacteria from viral infection. And so what does it do? Bacteria can create restriction enzymes, and these are like little scissors. And restriction enzymes are very specific, and they recognize genetic material, they recognize certain sequences within DNA, and they cut it up. And what they're targeting really is viral genetic material. And so the bacteria produce enzymes that break down viral genetic material. But then there is the modification part. And what happens is the bacteria create these pairs of scissors, and all they know is how to cut. They don't care where or what the genome belongs to. They will cut viral genome with that sequence. They will cut bacterial genome with that sequence. They would cut human DNA with that sequence. If it's got the sequence that the restriction enzyme recognizes, it's going to cut. However, the bacteria don't want to die in the process of creating these restriction enzymes. After all, the whole point is to protect them from viral infection. And so they have this modification system which protects their own DNA from the enzymes that they are creating. Now, we turn around as human beings, we've discovered these restriction enzymes and recognize that they can be really useful in biotechnology because we can use them to cut plasmids at very specific sites, and we can insert foreign DNA, and in that way, we can create recombinant organisms that can do things they couldn't do before. And so we can create bacteria that produce growth factors and all kinds of stuff. Another really interesting defense mechanism that some bacteria have against viral infection is this system called the CRISPR-Cas9 mechanism. And what's happening here is in some ways it's somewhat similar to restriction modification. There are scissors involved, but this is highly specific, once again, to certain genomic sequences. And what happens, the virus gains entry into the cell, bacteria produce this CRISPR system that cuts the viral genome, but here, instead of just cutting it up, they actually take a piece of it and insert it into their own genome so that if the virus ever gains reentry, they can make a copy of that, and that copy can bind to the viral genetic material and basically render the virus useless. CRISPR-Cas9 has been a really important discovery for us human beings because just like restriction modification, we've learned how to take this highly specific mechanism and use it in human beings to do gene insertion, to potentially block genes that are producing proteins that they're not supposed to be producing anymore, and to potentially correct genetic disease. The last process I want to talk about for bacterial defense mechanisms is a really ingenious one, and that is some bacteria have what is known as a toxin-antitoxin system. And what happens is bacteria are producing both of the toxin and the antitoxin at the same time. The toxin has a longer half-life inside the bacterial cell than the antitoxin does. This sounds dangerous for the bacteria, and it is. However, as long as the bacterial cell is healthy, it's producing the antitoxin to neutralize the toxin and everything is fine. But remember what we said earlier, when viruses infect bacteria, they usually take over the bacteria's ability to manage their own genome. And so the bacteria stop producing both the toxin and the antitoxin. Well, we already said the toxin has a longer half-life. So what do you think is going to happen to the bacteria? Eventually, it dies. But in doing so, it potentially saves the rest of the cells in the colony because the virus doesn't have time to get in there and replicate before the bacterial cell dies and prevents the replication from happening any further. We actually have something very similar in the human immune system, and that's something we're going to be talking about in a future video. All right, so this was just a brief overview of viruses and the study of virology. As always, thank you so much for being here. Have a great week, everyone.